Welcome, everyone. Happy Friday. Welcome to the Myotonic Dystrophy Foundation's Meet the DM Drug Developer Series. We're glad you could join us. My name is Tanya Stevenson. I'm the CEO of the Myotonic Dystrophy Foundation, and today we are going to meet with Dyne Therapeutics. Just a bit about the foundation. We envision a world with treatments and a cure for myotonic dystrophy. The mission of the foundation is community, care, and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatments and a cure. Just a reminder of the resources and support available from MDF. You can find our toolkits and publications on our website at myotonic.org/toolkits-publications. Our support programs, including all of our support groups, are available on myotonic.org/find-support. Our calendar of events, which includes all of the events happening for the foundation at myotonic.org/calendar/month. And our digital academy, which is all of our presentations, including presentations from the Drug Developer series, as well as Ask the Expert series, and our conference. Those can be found on our digital academy at myotonic.org/digital-academy. Today we have Dyne Therapeutics with us, but just a reminder, the first Friday of every month until our conference at 12 o'clock Pacific, we will be meeting with different drug developers. So if you haven't already, go to myotonic.org/meet-dm-drug-developers and sign up to hear from the rest of our pharma companies. So excited that we launched conference registration. We will be in Washington, D.C., September 7th through the 9th. We will have an MDF Advocacy Day on Capitol Hill on September 7th, and then our general conference programming will happen on the 8th and the 9th. Please register and learn more information at myotonic.org/conference. Today we are fortunate to learn from Dr. Ashish Dugar from Dyne. He has over 20 years of broad experience across medical affairs, clinical and commercial development, and real-world evidence generation. Prior to joining Dyne, Dr. Dugar served as Vice President and Global Head of Medical Affairs at Head Therapeutics, where he built the company's medical affairs organization. Previously, Dr. Dugar led all commercial and market access efforts for intracellular therapies as Vice President and Head of Commercial Development. Dr. Dugar also has held executive management roles at Roche and at Pfizer. He began his career at the National Institutes of Health and holds a PhD in pharmacology from Penn State College of Medicine. We are also delighted to welcome Ms. Molly White from Dyne and formerly from MDF. Molly is now the VP and Global Head of Patient Advocacy and KOL Engagement. She brings extensive experience in patient advocacy and community engagement in her role at Dyne, where she leads global patient advocacy and KOL engagement across all the company's disease focuses. She previously served as CEO of the Myotonic Dystrophy Foundation, a global patient advocacy organization. In that role, she led strategic direction and administrative oversight for the comprehensive programmatic and fundraising efforts of the foundation and its staff. She has over 25 years of leadership level experience building, implementing, and running award-winning domestic and international community programs for organizations in both the private and nonprofit sectors. She received her master's from the University of Iowa. Many of you submitted your questions live. Thank you so much. I'm sorry, in advance. Thank you so much. But you can submit questions live during the presentation. Most of the Q&A will happen at the end of the presentation from Dyne. They have done their best to integrate answers to the questions that were submitted in advance. They have a presentation that should address most of the questions submitted in advance, but in the event you have additional questions, please don't hesitate. Just go to the Questions tab, type in your question, and click Send. If you're on a smartphone today, just click the question mark icon at the top of the screen, type your question, and click Send. If today's presentation speaks to you, please consider a gift to the foundation, myotonic.org/donate. Okay, it is my pleasure and privilege to introduce the Dyne Therapeutics team. Welcome.
Great. Thank you so much, Tanya, for that phenomenal and flattering introduction for myself and Molly. As Tanya said, I'm Ashish Dugar, the Chief Medical Affairs Officer at Dyne Therapeutics, and joining me is Molly White, someone very well known to this community, and also Dyne's Global Head of Patient Advocacy. First, we'd like to thank Tanya, Clede, and the entire MDF team for inviting us to present at this webinar to update the DM community on the progress of our program, and specifically Dyne 101, our clinical candidate designed to treat adult patients with myotonic dystrophy type 1. As always, it's a true privilege to speak with the myotonic community. So thank you very much for your time and attention and all of the questions that you've already submitted, and we will definitely be looking forward to more questions at the end of this presentation. These are our forward-looking statements. Please refer to our website if you are interested in more details. So at Dyne, it all starts with our mission. We're laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with DM1 being a priority. Our mission is core to who we are and what we do every day. We're really excited to inform you that we started dosing participants in our Dyne 101 clinical trial called Achieve. And our focus today is on sharing an update on our clinical trial progress and details about our clinical trial with Dyne 101. So we're building Dyne as the leading muscle disease company to deliver for families living with DM1 and other muscle diseases. Shown here are 3 core strengths to help us deliver on that. First is our commitment to delivering for patients. We've initiated our clinical development program in DM1, which has been informed by the patient community and scientific leaders in the field. Second, we have exceptional in-house and advisory expertise with deep knowledge of muscle diseases such as DM1 and experience in developing novel therapeutics. And we have a world-class scientific advisory board, which includes luminaries such as Dr. Charles Thornton. Third, we intentionally designed Dyne 101 to overcome one of the biggest limitations of potential therapeutics, which is effectively and efficiently delivering modern oligonucleotides to muscle tissue. Dyne 101, our clinical candidate, is designed to reduce toxic nuclear DMPK across key muscles and correct the splicing defects to potentially stop or reverse disease progression. So I don't need to tell this community much about this particular disease. As you know, DM1 is an autosomal dominant progressive multi-system degenerative neuromuscular disease, and it's most commonly diagnosed in adulthood. But we know symptoms can appear anytime from birth to advanced ages, depending on the subtype of myotonic dystrophy type 1. Symptoms include muscle myotonia, which is the inability of muscles to relax after using them, often described as muscle stiffening, followed by weakness and atrophy of skeletal muscles. Cardiac conduction abnormalities and respiratory function distress is also part of this disease. Importantly, gastrointestinal and metabolic disturbances affect quality of life greatly, as well as fatigue and sleep issues. And together, all of these greatly impact the quality of life for the individual and their families. And we recognize that for a rare disease, DM1 is not so rare, and it often impacts multiple generations within a family. So this slide describes how proteins are made. So a little bit of biology 101 here, which I think is helpful to know to better understand the cause of myotonic dystrophy. So cells, as I'm sure everyone knows, are the building blocks of our bodies and responsible for many functions. And DNA is a substance in the cell nucleus that contains the instructions for making proteins which do the work in the cells. Now, a section of DNA with instructions for a particular protein is called a gene. And that DNA information about genes is written into RNA, and it's a process called transcription, which is then cut or spliced into something called messenger RNA or mRNA. And then that messenger RNA or that mRNA leaves the nucleus and translates the DNA into proteins. So this is a pretty, brief schematic of how proteins are made, which is foundational to helping us think through what needs to be done as we tackle myotonic dystrophy. So I won't go into all of the detail on this slide, but hopefully it provides a useful reference. Briefly, this shows that the defect in the DMPK gene causes myotonic dystrophy type 1. Now, it's known that DM1 is caused by high numbers of abnormal repeats in a DNA section of the DMPK gene. Specifically, people with DM1 have abnormally high repeats of CTG, and C and T and G are building blocks of DNA, and we see these abnormally high repeats both in the blood and the muscle tissue. So what happens is that the CTG repeats are copied to make RNA, and abnormally long CTG repeats make toxic mRNA that trap proteins in the nucleus. And these trapped proteins are needed to process RNA in the nucleus through a process called splicing. Now, these trapped proteins form clumps, if you will, made up of mRNA and proteins that can't leave the nucleus to do their job in terms of splicing. And so this leads to abnormal splicing. And it's this abnormal splicing that's believed to cause many of the symptoms of myotonic dystrophy type 1. So currently there are no disease-modifying treatments available. And so right now the management of DM1 is limited to managing the symptoms. To treat the genetic basis of the disease, the key is to get the therapeutic agent into the nucleus of the cells and knock down nuclear toxic DMPK RNA, which can then correct the splicing defect, which may lead to slowing or stopping the progression of disease. And Dyne's targeted delivery approach is designed to effectively and efficiently deliver our therapeutic agent to muscles and patients so that we can make a meaningful difference to patients' lives. And as I go through some of our preclinical data in a few minutes, how our investigational medicine works will become more clear. So here I'm going to spend a few minutes to describe the FORCE platform. The FORCE platform drives our efforts to develop and deliver muscle-targeted therapeutics. It's expressly designed to address that one major issue in muscle diseases, which is to efficiently and reliably deliver antisense oligonucleotides that target the genetic basis of diseases and getting to those important muscles like skeletal muscle, including diaphragm and respiratory muscles, cardiac muscle, and smooth muscle. As you can see, Dyne's FORCE platform consists of three parts: an antigen binding fragment, or what we call a Fab, that enables us to have targeted delivery of the ASO to muscle, a clinically validated linker, shown in the burnt orange string here, and an antisense oligonucleotide, or an ASO, selected to target the genetic basis of disease. Now, I'll start by describing these three components and then come back to the importance of how we target our therapeutic agent using the transferrin receptor 1 or the TFR1 to be able to drive targeted delivery of DYNE-101. So we chose a portion of a monoclonal antibody or an antibody fragment or a Fab that binds tightly to this TFR1 receptor that I'll come back to. Now, we chose this piece of the monoclonal antibody or this Fab because we believe it may offer powerful advantages over the larger monoclonal antibody, including its smaller size, so ability to get into the cell, its lower overall protein load, and the potential for enhanced tolerability profile. As I've mentioned a few times, the goal is to ultimately enable targeted, effective delivery of the therapeutic molecules to skeletal, cardiac, and smooth muscle. We selected the Val-Cit linker for our platform based on its stability and clinically validated safety and efficacy in approved products. There are approved products in the US that use this linker, and we believe this further de-risks our overall platform. Now, we're utilizing the same Fab and the same linker across all of our programs. So, for example, our Duchenne program, which is also in the clinic. The third component is the ASO chosen to target the genetic basis of myotonic dystrophy type 1. So where disease-modifying nuclear knockdown is key, as is in the case for DM1, it's critical that we select an ASO that's preferentially trafficked to the nucleus. And so for this reason, we designed an antisense oligo that targets the toxic DMPK in the nucleus. Now, the mechanism of force is different from other approaches. We're essentially using the cell's natural biology to deliver our therapeutic to muscle cells. Our Fab binds tightly to something called the TFR1 receptor, as I mentioned earlier. Well, TFR1 receptors are highly expressed across the surface of muscle cells, including skeletal, cardiac, and smooth muscles. So we're able to harness what already exists on the surface of muscle cells to deliver our investigational medicine through this receptor-mediated mechanism. And by using TFR1 with Dyne 101, we should have the potential for a wide safety margin and durability of effect, and also be able to dose relatively infrequently. And I'll talk more about this as we get into the clinical program. So the next few slides highlight critical studies we did and key data we generated before going into the clinic. We needed to do quite a bit of work to make sure we felt that it was the right time with the right data behind it before we ended up in clinical trials. So to prepare for that entry into the clinic, Dyne has deliberately conducted comprehensive preclinical studies across our programs. And we've not shied away from asking the tough questions or running the right studies to evaluate our investigational medicine. We've operated with a strong desire to understand our molecules and best position them for safety and success in patients. The team has generated the most comprehensive, or among the most comprehensive, significant and robust data sets across programs that we believe sets a new bar in our field. Whether it's in vitro patient cells, various mouse models to evaluate pharmacokinetics, meaning how it moves, how the force conjugate moves throughout the body, pharmacodynamics, essentially looking at DMPK knockdown and splicing, as well as function. And we've also looked at non-diseased, non-human primate or monkey studies to understand the distribution of our investigational medicine to muscle, as well as safety and tolerability. I do want to point out on this slide the importance of the HTFR1 DMS XL mouse model. And you can see that here in sort of the middle bar. The literature indicates that toxic human DMPK RNA is the genetic basis of DM1, and it resides in the nucleus. So we set out to conduct our own experiments to understand this further, as well as the ability of Dyne 101, our investigational medicine that we're taking into clinical trials, to reduce this toxic nuclear DMPK. So we developed this HTFR1 DMS XL mouse model by crossing mice with the knocked-in human transferrin receptor, and these mice already carry the human mutant DMPK transgene with over 1,000 repeats. So what does this mean? Basically, this mouse model expresses both the human transferrin receptor and human toxic DMPK. So we can test our actual clinical candidate, Dyne 101, in this mouse model. And this is important because what we find from these animals can help us understand the translatability to patients. So when we evaluated Dyne 101 in the heart of the homozygous HTFR1 DMS XL mouse, after just 2 doses of the relatively low dose of 10 milligrams per kilogram, we observed a reduction in foci, so sort of those trapped proteins I was talking about, and the toxic DMPK at about 49%. And this resulted in compelling splicing correction at 4 weeks that you can see on the right-hand side of the slide. And we know that cardiac muscle is difficult to access in muscle diseases, and this is critical to improving the condition of patients. We have seen similar results across other key muscles involved in DM1, including skeletal muscle, diaphragm, and we've also explored smooth muscles. We're really excited by the data from the numerous studies using this mouse model as well as other studies, and we believe this sets a new standard for evaluating DMPK knockdown and splicing in DM1. So importantly, we also utilized non-human primate or monkeys to evaluate our ability to get into muscles of a larger species and engage that target, namely the DMPK, and assess safety and tolerability. It's important to note that monkeys are, this monkey model is not a disease model, they're healthy, so we're not looking at splicing. And in one of our NHP studies, we demonstrated that Dyne-101 robustly reduced wild-type DMPK in multiple skeletal, cardiac, and smooth muscles. These data demonstrate that we can deliver and engage the right target in large animal muscle tissue. We also evaluated monkeys in a 13-week GLP toxicology study. And as shown here, we were very pleased that Dyne-101 was safe and well tolerated after repeat dosing. We observed no dose-limiting toxicity and no changes in cardiac, respiratory, or kidney function. So all of this data gave us a lot of confidence to move into the clinic. We've also done extensive work to prepare our entry into the clinic. We had engaged with stakeholders around the world as part of this effort. This engagement has included advocacy groups such as MDF, patients, and key opinion leaders from various disciplines, including neurology, cardiology, physical therapy, pulmonology, and other areas to ensure that we have their crucial perspectives on how to move forward with our clinical program. These thought leaders, with their tremendous experience as clinicians in running clinical trials, have provided us with critical input on the overall design of the clinical study, biomarkers, endpoints, including splicing, myotonia, various strength and functional outcomes, as well as key safety considerations. And they've also provided key learnings from natural history studies. We've also reached out broadly to advocacy leaders, patients, and caregivers, many of whom are healthcare providers themselves, to ensure we have a patient-centric study design, and provided advice in a number of areas, including clinical trial assessments, placebo and biopsy-related considerations, as well as clinic visit designs and decision-making insights for the selection of even participating in a clinical trial. And we're also leveraging insights from the NDM1 natural history study as we designed our clinical program in DM1. I'm sure, as many of you know, it's one of the largest and most comprehensive natural history studies conducted to date in DM1 and have support from a number of institutions and companies. And this has provided us great learnings as well. So our clinical trial for Dyne-101, called ACHIEVE, is a global trial consisting of essentially 4 periods. We have a 6-week screening period, followed by a 24-week placebo-controlled period, a placebo-controlled multiple ascending dose period, where individuals receive either placebo or Dyne-101 administered every 4 or 8 weeks. And this is followed by a 24-week treatment period where all participants will receive Dyne-101 administered every 4 or 8 weeks. And then finally, a 96-week long-term extension period. It's important to note that following the placebo-controlled portion of the study, all patients will be able to receive active treatment. We plan to evaluate many outcomes, including safety and tolerability, splicing, pharmacokinetics, and pharmacodynamic information to understand how the body is processing Dyne-101 and how it's acting in the body. And we'll be looking at several measures of muscle function and strength to help us understand change in how the body is functioning. And we also carefully are assessing lung and heart function. With the help of all of the expert advisors we've engaged, we're really confident that our study design is robust is going to measure key outcomes and is striving to manage the burden on families. And I'm very excited to say that we'll be communicating some initial safety, tolerability, and splicing data in the second half of this year. So here you can see some of our inclusion and exclusion criteria that must be met for an individual to participate in our study. For more information on the study design, inclusion exclusion criteria, and open trial sites, please visit our ct.gov posting and you can find the number listed here. Just very briefly, we're enrolling approximately 64 individuals between the ages of 18 and 49 inclusive. We are ensuring that there is a definitive diagnosis of myotonic dystrophy type 1. As I mentioned before, this is about adult onset myotonic dystrophy, and so onset of DM1 muscle symptoms is starting at least at 12 years of age. And then we have other measures of muscle function and strength to make sure that participants are qualified to be in this study. And we do have a number of open sites around the world, and you can see some of those countries listed here, including New Zealand, France, the Netherlands, UK, and Italy. And we continue to open up more sites. And finally, I think it's really critical to thank you. We couldn't do our work without your support, encouragement, and sacrifice. We understand that work from countless dedicated scientists, physicians, physical therapists, and other healthcare providers across the world is needed because myotonic dystrophy is so devastating with no approved treatment options. And then we know that the patient community is making huge sacrifices to advance the science as well. And we want to serve all of the community, such as people listed here like Suzette and Billy Dean. So thank you very much for your time and attention. We greatly appreciate it. So for this next part, we'll go through questions and answers that you've provided already. Molly White is going to facilitate the Q&A, and I'm going to hand it over to Molly.
Excellent. We are so delighted to chat with you all today. So we got a bunch of very good questions submitted through the team at MDF before the webinar this morning, and we'll start by answering some of those that we think get at the bulk of the questions that you all had. And then we can take some of the questions that you send in over the course of the webinar today via the questions link. So maybe, Ash, you can start with the first one, which is maybe you can talk a little bit about the difference between Dyne's therapy and other therapies for myotonic dystrophy type 1 out there.
Sure, happy to. So hopefully I hope the presentation that I did on the Force platform provided some good insight into our mechanism of action and some of the differences between us and others. So I'll reiterate that we intentionally designed Dyne 101 to knock down that toxic nuclear DMPK using an antisense oligonucleotide that we designed. And this is to correct the missplicing that underlies the functional disability associated with DM1. DM1 is a spliceopathy. As I mentioned, we use a portion of the monoclonal antibody that binds to the TFR1 receptor. And this is different in the way we work versus how others work. Second, we use a clinically validated linker, so a linker that's been used in other programs, in approved products. And again, that's another difference between our investigational medicine and others. And finally, we use this, as I mentioned, the antisense oligonucleotide. ASOs are agents that get into the nucleus. So we wanted to make sure that we were attaching something to the Fab, to our Fab, using the linker that would get into the nucleus where we need to knock down the toxic DMPK to affect the splicing to change function. So, our three main components of Dyne 101 are differentiated from other compounds that are out there. This is designed to be disease modifying, so it's really designed to change the course of the disease and not just be some symptomatic management. And we've been able to show promise using the animal models, and hopefully this translates as we're generating clinical data.
Excellent. Another question we had was, when are we coming to the US and why didn't we start in the US? So we want to assure everybody in the MDF community that we are committed to bringing Dyne 101 to the US. And what we're currently doing at the moment, as you can see from the clinicaltrials.gov site, is we are generating data outside the US, and then we will bring it to the FDA for a discussion on how we can bring the ACHIEVE study to the US. And as we have more clarity on what that process is going to look like and timing, we will continue to update all of you as soon as we can, working with MDF and others. Let me also have Josh or Ash, maybe you can jump in on this one. What do we hope to see from this trial? You know, will it impact skeletal muscle? Will it also work in cardiac, respiratory, or CNS? We'll also attack those symptoms as well.
Yeah, it's so really good question. So let me try to tackle it in a couple of different ways. So I've shown just some of the data we generated in our preclinical animal studies. As part of the data we've generated in the HTFR1 DMS XL mouse model, I showed the ability for Dyne 101 to enter the heart and positively affect the toxic nuclear DMPK knockdown, reduce those clumps or that toxic foci, and correct splicing. We've done other work not shown here in a different mouse model that actually has a myotonic phenotype, for example, and we've been able to show improvement in that myotonic phenotype. And we've also looked at this in monkeys. So we're really excited about the full data package. And what we also did in the HTFR mouse model is we looked carefully at skeletal muscle, diaphragm as well, and we showed a similar pattern of response. So we feel like we're able to demonstrate in relevant animal models impact on many of these areas that have been asked around skeletal, cardiac, and respiratory. And we're certainly committed to addressing all aspects of the disease across all individuals with myotonic dystrophy type 1, including congenital and including other aspects of DM1, including the CNS symptomology with Dyne 101 and future agents. So we're continuing to advance our program, and we'll keep the community apprised as we generate more data and advance our clinical trial program. I would say, the CNS questions come up a few times. Importantly, we know there are many companies that are using the TFR1 receptor to take therapeutics across the blood-brain barrier to try and alleviate various disorders. And it's really encouraging to see companies go down this path. And this provides learnings for the field, for us, and so it's going to be important information that these companies are generating. As a reminder, our study is evaluating, this particular study is a safety and tolerability study that's evaluating adult onset myotonic dystrophy type 1. Currently, we're not studying other classifications such as congenital or childhood onset. And I think there was another question around what are other companies doing? And I think, again, the ct.gov website is a great place to get some information. MDF is a great place to get information on what companies, are doing. That said, we recognize there isn't, enough clinical development work going on in many of these other subtypes, but I do expect that this changes within the next few years.
Excellent. We had a question, and we often get this question, will our therapy or our potential therapy work in DM2? And as this community knows better than anybody, DM1 is a CTG expansion in DMPK, and DM2 is a CCTG expansion in CMPP. So it's a similar autosomal dominant muscle disease caused by genetic changes, but there is a difference between DM1 and DM2 that's significant enough that it would require a different, ultimately a different therapy. So they're distinct disorders. They'll require different therapeutic approaches. I can say that Dyne is very committed to delivering potentially transformative therapies for as many people in the DM community as possible. And I think, we will continue to move our exploration forward. And when we have updates, as always, we will share them with MDF, and we'll get them out to the community as soon as we can. Ash, do you want to talk about when we're going to be able to report on some data?
Yeah, happy to. So, as I think I mentioned, and as we've put out there, we anticipate sharing initial clinical data from ACHIEVE in the second half of 2023 in the form of safety, tolerability, and splicing. the entire duration of the ACHIEVE study, as you know, it's a 3-year study. Obviously, the study takes longer to fully complete, but any given participant is in the study for up to 3 years. But we're really excited about sharing some data at the end of this year.
And then we had another question, sort of a series of questions around how many people are participating, who's eligible to participate in the ACHIEVE study, where are the sites going to be located, is there a site in the UK? So just generally, as we've said already on this call, ACHIEVE is a phase 1/2 clinical trial. It's evaluating Dyne 101, which is a potential therapy for people with myotonic dystrophy type 1. It includes a 24-week multiple ascending dose randomized placebo-controlled period, followed by a 24-week open label extension and a 96-week long-term extension. And so as Ash has said, the total trial length is about 3 years. We're currently enrolling around 64 adult patients between the ages of 18 and 49, although we may be adding additional cohorts and expanding the total participant count, as we go forward. And as we've said, ACHIEVE is a global study. You can go to the clinicaltrials.gov site and you can see the multiple countries that are currently enrolled. We will continue to add to those as we bring more sites online. And to the question around the UK, we do have sites in the UK currently enrolling. So we have gotten a couple of additional questions through the questions chat. Ash, maybe I'll toss this one to you. Someone was wondering, are there other cell types that we use to assess Dyne 101 beyond muscle?
Are there other cell types beyond muscle?
What patient? Yeah. What patient cells were used in the assessments or the studies? Was it just muscle, or were other cell types also used?
Oh, great question. Yeah. So in the in vitro experiments that I just touched on in sort of that one slide listing the kinds of preclinical work we did, they were patient muscle cells. So the in vitro work was only in muscle cells.
And then we have a question, what exactly is splicing? Do things need to splice to make the muscle stronger?
It's a good question. So it's essentially a mechanism that the body uses to be able to ensure that there is the proper formation of proteins that are important for muscle function. So when there are in that sort of RNA, you want to be able to cut away certain pieces so that you have ultimately something that comes together properly, is read through properly by the body to be able to form the protein, sort of that translational process. And when that can't happen, you have the body sort of go haywire with the missplicing, and then the body is unable to produce what it needs to produce for proper muscle function. We could probably have a whole hour-long discussion on splicing here, but many of these muscle diseases, such as DM1, even Duchenne, which is a toxic loss of function as opposed to myotonic dystrophy, which is a toxic gain of function kind of disease, there is abnormal splicing associated with these muscle disorders, and being able to correct the splicing is absolutely critical to be able to alleviate the disease and modify the disease progression.
We've got another question here. Do you think that this therapy, Dyne 101, will potentially reverse current damage in the muscle cells?
Yeah, that's a good question. The one way to answer it is, we'll find out what Dyne 101 can do in terms of muscle damage as we generate data from our clinical trial. What I would offer is that unlike muscle diseases where muscle is replaced by fat and fibrosis, so it's much more difficult to recover, sort of gain that function back. We're hopeful that in myotonic dystrophy that there is the potential to actually reverse the muscle disability, muscle symptoms. So, we've talked about this a few times, but certainly we want to be able to stop any further progression. And we are also hopeful that we might be able to reverse some of the symptomology.
There's a question regarding whether the trial will include older adults at some point.
So I think we get a lot of questions on, will the trial include younger patients, older patients? Will you expand it in this way or that way? I think it's really, not to get into a sort of a drug development discussion here necessarily, but this is a phase 1/2 safety and tolerability study. Given that this is the first time Dyne 101 is going into individuals, we want to proceed very carefully. The first tenet is, do no harm. We want to be able to understand that this drug is safe and tolerable, and we'll be measuring muscle function and strength and other aspects. But we want to really tread carefully right now, have a more uniform, homogeneous patient population so that we can understand very carefully how safe and tolerable this drug is. From there, we can then expand, whether it's this trial or other clinical trials with Dyne 101, but we really need to be careful in understanding what this can do in a relatively homogeneous population.
So I think there's a couple here that are pretty interesting. I think a lot of people are probably wondering about this. One of them is, as the trial advances, will you be allowing people to join in later stages? So we can talk about the whole cohort structure. If you determine that phase 1/2 safety, tolerability, et cetera, is good, can a person jump into the study in phase 2 or 3?
Yeah, it's a great question. So, right now, the ACHIEVE study is designed in the way it is, with conversations with regulatory authorities around the globe. And then for us to change the design or enhance the design over time is something we are always evaluating, because we want to be able to learn as we go along and make sure that we are thinking about the right trial design. And so we'll continue to do that and have conversations with regulators on any changes that we might want to make. That said, we certainly understand this is not going to be the only clinical trial with Dyne 101. We are going to do other clinical trials with Dyne 101. So there will definitely be the opportunity for expanding access in terms of through this, through the clinical trial program to Dyne 101.
I think this is also a really good one. So there's a question here of the 64 participants that we're currently identifying, and we did point out that there may be an expansion in the total number. Do we expect all of those participants to be recruited outside the U.S.?
Yeah, no, I think as Molly mentioned, we are certainly committed to bringing the Myotonic program here to the U.S. So again, as Molly said, we're going to be looking to generate some data and have a conversation with the FDA on the best way to open it up in the U.S.
I also think, I have to say, you guys are doing a great job. These are really interesting questions. There's a question here around, since DM is multisystemic, as we all know, will Dyne 101 potentially rescue non-muscle DM features?
So I think that, again, this might be alluding to, I don't know if it's alluding to CNS, but we are targeting the muscles, as I've mentioned, including smooth muscle, which we haven't talked a lot about. So Dyne 101 in this trial is really focused on looking at safety, tolerability, and muscle symptoms, strength, function, and we have patient-reported outcomes in here as well. So we are also looking to understand how the patient is feeling, and being able to understand things like fatigue or other areas. So we really want to be able to understand how this is working with the patient holistically, and that is what this current study is focused on. As I said, we are certainly interested and need to think about how to serve the entire community across the full spectrum of symptoms. And it's great that other companies are also looking at non-muscle areas so we can learn from them as well.
A lot of interest in the U.S. component and when we're going to launch it. There's a question here. Will your next steps in the U.S. follow completion of the international portion, or will it be concurrent?
So, sorry if I'm going to sound like a broken record. So again, we are going to look to generate data from outside of the U.S. In the current sites that are enrolling, and then have a discussion with the FDA and think about how we can bring this to the U.S. So that's what we're going to try to do for this particular study for Achieve.
And I think we have captured all of them. There are different, there's some additional questions around CNS. I think we've addressed what we can there. I think we've got it.
Wonderful. Great questions. Really good questions, tough questions. That's great.
Thank you so much, Molly and Ash. This was fantastic. Thank you for the presentation and really the education, especially the clarity around your process and the therapeutic itself. I also want to thank you on behalf of MDF because you are a fantastic diamond sponsor of our upcoming conference. And we are thrilled that you may have some data to share with us for Achieve at the conference in September, it sounds like. So we'll keep our fingers crossed. Thank you both for being here today and sharing with us. And for all those listening, thank you for being here and have a fantastic weekend. Take care, everybody.
Bye, everybody.
All right. Thank you.